Okay, today is April the 16th. We're doing the urinary system, and we've got a picture up here on the screen of the kidney. So, this is a histology class, so there are lessons to be learned about structure and function, which is what is unique about this course and separates it from just physiology. But you guys remember the kidney, right? It's an organ dedicated to filtration. When I was in graduate school, we would do a lot of experiments with fluids, and they were going through chromatography and that sort of thing. And then we came to the conclusion that a big part of life was about the plumbing. So here we are. So it's not just the circulatory system, but it's the plumbing of fluid in the body. And it matters how that fluid is handled, and it has to be handled exactly right. It is about the plumbing. So urinary tract is not a minor thing. It's purifying the blood. And as we've talked about in the circulatory system, there's let's see. The blood supply is carrying nutrients and, of course, carrying oxygen to various parts of the body. But it's also picking up CO2, and it's picking up waste products. And so the body's got to get rid of that. So the kidney is involved with, of course, filtering out those impurities, and you're going to end up with urine eventually that you're going to send down from the kidney, and you're going to hold it in the bladder until it's expelled. But it's more than just that. You need to control the fluid level, the pressure of the fluid level, and there's quite a bit to the whole thing. Okay, so let's look at the kidney then. So there are these different areas to the kidney. The outside, besides there being a fibrous capsule around the whole organ to protect it, there's the renal cortex. And the word renal, you should know that. So if there's a test question and it says something about renal, we're talking about kidney. And sometimes even a little piece of information like that will allow you to answer a question correctly. So the renal cortex is closer to this fibrous capsule on the outside, and the renal medulla is just the next step in from that. But the cortex goes all the way around, right? You see it's sort of continuous. Whereas the medulla is, it's separated into these separate units, which the separation is through this renal column. Sort of looks like a fruit, doesn't it? You know, like you slice open one of those exotic tropical fruits or whatever, and so there's these different, there's a structure. But these things, the renal pyramid is just a way of saying, okay, this is kind of, wider here, and then it narrows down, and it's narrowing down to where it's going to feed into these calyx, the minor and major calyx. And these calyxes, this is where the urine is going to go. It's going to the ureter. And the ureter is not the urethra, which is the one we're always thinking about, because that's the one, that's the one that you're excreting out of. But the ureter is the tube that's just bringing this urine to be held by the bladder. So the calyx is the part of the ureter that is going into the kidney itself. And then they have names for these different things. Like overall, this hilum here is the dimple that's in the middle of the kidney. And the renal pelvis is this thick area just as you transition from the ureter into the kidney itself. So the basic unit of the kidney, though, is the nephron, and I think we've all heard about that in basic biology classes. And there's a bunch of them. There's a million of them. So we're going to discuss what that looks like, because this is histology, not gross physiology. So we're interested in what's happening microscopically here, but it is useful to get a cross-sectional view of the whole organ itself. So this filtration, these nephrons are both in the renal cortex and in the renal medulla, and you'll see how that separates out. But they're present in both. But the renal medulla is going to be closer to these calyxes that are dumping the urine into the ureter. Okay. Okay, here we go. So this is the basic unit. This is the nephron. And somebody commented to me that it would be good to know what the vocabulary words are that we need. So first thing that I mentioned was renal, so relating to the kidney. That the basic unit of filtration is the nephron, and we're looking at a diagram of a nephron right now. Also, the renal, you should know the cortex, medulla. Okay, so the nephron is a fairly small structure, and that's where you can actually see morphologically what's going on. So when we look at some pictures of areas within the nephron, we'll see, what do you think we'll see? What kind of cells would you expect to see?
In the nephron?
Yeah. So if you're slicing the nephron in various angles, what do you think you're going to see?
Epithelial cells.
Okay?
Epithelial cells.
Yeah, lots of epithelial cells. So this is mostly about epithelial cells. Of course, there's a lot of vascularization, so you're also going to see what kind of cell? Not kind of cell. You see vascularization. The vessel. A capillary has to have at least one kind of cell that makes up those tiny capillary tubes. What is that cell? Put you on the spot, didn't I? It's an endothelial cell. So endothelial cells are, I think that they're related to epithelial cells in some ways, but there's mixing between epithelial and mesenchymal cells. They can transdifferentiate, we've learned, at some rate. There's the epithelial mesenchyme transition, and then there's actually mesenchyme going to epithelia as well. So these things are not as narrowly defined. They're not committed to a certain type of differentiation quite like we had envisioned years ago. It's much more fluid than that in terms of what they're capable of. But anyway, so we're dealing with lots of epithelia. So the pictures are going to show us lots of epithelia. What do epithelial cells look like? Right? Are they flattened? Is there a lot of material in between? What are you used to seeing for an epithelial cell? Well, hopefully you're thinking, well, epithelial cells, they're, the cuboidal thing. They're stacked next to each other, something like that. They're not flattened. So, that's a fibroblast, a connective tissue cell. So you don't see that. Although squamous cells can be more flat. Differentiated epithelia can be flattened down. But in the kidney, we're not looking so much at a differentiation, right? We just need a simple epithelia. So this is a simple epithelia. So this is really what we're talking about. Some kind of single layer of simple epithelial cell. And not stratified cells and connective tissue cells so much. Okay, so how does this all work? So you see that you have a blood supply, and the blood supply is it's in this glomerulus, and that's an important word. You have to know that, of course. So where's the eraser? Okay, so this is kind of what you'd expect to see, right? Because there's got to be a ball of these tiny vessels. And the reason why you're doing that is because you want to optimize the surface of exchange. So you're trying to get urea and waste products out of the blood. You're filtering the blood. So you create this capsule-like structure here. So they call this a capsular space. Is the glomerulus overall is the word used for this entire structure, but it's also the individual folding of these vessels, and this space, capsular space here, is an area where anything that's filtering out from the microvasculature can be taken away. And it's taken away by a set of convoluted tubes, tubules and tubes. The tubule is proximal, distal, loop of Henle, which I think everybody's heard of that before. Why is that so important? It has a proximal, descending and ascending limb. It gets thin down here and then it widens up again up here. Why is this important? Why all this tubing important? Remember I said that life boils down to plumbing. So we're looking at the plumbing, a complex diagram on a microscopic level of some very important body plumbing. And so it was important to have a lot of the microvessel kind of compacted into this sphere, into this corpuscle, because we wanted to optimize the exchange across these vessels. But once you've got stuff that comes out of that, why do you need so much plumbing here? Why all this complexity?
It's just for more filtration to occur.
Yeah, it's more filtration, but what do you need to filter? I mean, you're filtering stuff out of the blood, right? The bad stuff out of the blood. So stuff is coming out, and then so why do you need to filter more?
So stuff could go in.
Yeah, right. Yeah, because only in the glomerulus do you get the movement of these solutes out of the blood. So the rest of this system here is not. It's not part of that system to pull stuff out of the blood. It's a system designed to put stuff back in. So what kind of stuff you're pulling out and what kind of stuff is going back in? Just throw out some ideas.
Water's going back in.
What's that?
Water.
Water. Yeah, water is a big deal, and controlling the whole pressure of the system is a big deal too, and sort of coercing the water to come out of the blood is important too. So there's a little bit of squeezing that goes on in this whole process too, to try to pull water out and to put water back. And depending on how much water your body needs, the body can control that. Do you remember on the endothelial system we were talking about the diuretic factor? So there's a hormone that will actually, that's invoked in order to, tell your body how much water you need. Do you want to conserve water, or are we trying to get rid of water? So, these systems get to be a little bit complex, but let's think about it still in terms of plumbing. So, so water is going to come out. What else is in the blood? Yeah, so there's ions, right? So ions, also known as electrolytes. So yeah, ions, electrolytes. So you have to conserve that too. So if they come out, you want to put them back in. But on this filtration, what size are we thinking about here with all this stuff? This is one of the coolest things I think about working on a cellular level and tissue level, and then trying to understand physiology, is because at least you're at the right size that you need to be. And the filtration is a great example where the cells are small enough that they can actually filter molecules. These little blebs and things you see on cells, they can make openings that are too small for a large protein to go through. So, and they can actually have effects on ion concentration even, which is really amazing. Because you usually think of ions being, about as small as you can get. But the cell is the right size to deal with molecules, and that's why, by us looking at cells, we can actually get to the point where we can understand how this whole thing works. So let's just jump ahead for a moment, just to see what some pictures look like. We'll go back to it too. But here you have the diagram of the glomerulus here, and then here's a picture of what a single renal corpuscle looks like. But this thing that we're looking at these capillaries. They're fenestrated. What does fenestrate mean? Holes. There's holes punctured in these capillaries. That's pretty amazing. You think of, I was just saying that one of the things that's really cool about thinking about structure function on a cellular level is the cells are so small that they're small enough to be able to control molecule size. So a fenestrated capillary, it's similar to what we can make synthetically. We're only learning how to make filters now. There's a company, Millipore, that was specializing in filter membranes. And there's been various companies along the way. They were just bought out 2 years ago or so by EMD. So they're making these membranes that have these small pore sizes. But nature has already set this up, this system where you're not just relying on diffusion through an intact capillary, but actually a fenestrated capillary. So there's certain molecular size that can go through. Now that would be kind of a cool trick already, but then there's this other thing, these podocytes. And podocytes, kind of that picture looks like an egg or something, right? Because there's this nucleus here. But it's sitting on top of these capillaries. And it has a unique structure. It's forming these little branches that they call pedicels. And the pedicels are, I was holding my hands together here, they can control a slit size, down to the molecular level, pretty much, with the filtration slits formed by these interdigitating pedicels that are part of the podocytes that are sitting on top of fenestrated capillaries. A lot of filtration here, and the size is right, for being able to control what kind of molecules are going through. This is an impressive looking scanning EM. It's one of the, it looks like the diagram is pretty much, an artistic rendering of that scanning EM, but it's pretty impressive that if you look at the surface characteristics, it looks exactly like their picture here. I mean, that's how good, that's why it's so great to have these scanning EMs and the transmission EMs too, so that you can really see the fine structure, and it helps you to understand what's going on. So, in the case of the urinary system, we're dealing with something that is, it's about the plumbing, and it's about filtering, and it's about distinguishing different types of molecules and being able to control what kinds of molecules are absorbed versus what kind of molecules are going to be put back into the blood. What do you need to conserve versus what do you need to get rid of? And so, anyway, so I was just commenting that the scanning EM is really beautiful, and you see these pedicels. So there are these slits that are formed. So, dealing with these holes and then slits, you've got two different types of membranes, one over the other, that you can really control what size kinds of molecules are coming through that. The standard section of the renal corpuscle, it looks a little bit sloppier because there's, you can kind of make out some of the glomeruli lists there in this corpuscle space is obvious, but it's not that spectacular as compared to the scanning EM. And then there are these transmission EMs. So these are the pedicels. See how small they are? It's just a different way of looking at them, but this picture shows the same thing over here. So you see that they're kind of regularly spaced, and there's a certain size here that you've created for passage of molecules. So you see, it almost looks like a microcilia or something like that, but it's not. It's on a very fine cellular level. Okay, so let's go back. It's not going back, is it? No, it doesn't go back. Okay, so this balance between secretion and reabsorption is taking place in this basic unit, the nephron of the kidney. And the nephrons are contained both within the cortex and the medulla. And depending on where they're positioned, the size of this loop can change. So the way they've drawn the diagram here, here's the cortex, and then here's the medulla. But depending upon where this nephron is, you can make this loop of Henle longer to be able to extend into the medulla if it happens to be higher in the cortex, or there's actually some of these that are right in the medulla. And so, I don't know, do you want a list of things? So urine is the thing that we're trying to excrete. So, so of course that's the main objective. right? But then you also want to keep the amount of water correct to have the right kind of blood pressure overall. If the water level got too low, that would tend to reduce blood pressure. Okay, let's see, here's a diagram. So, I have a few. So here's the glomerular filter, and we had just talked about the fenestrated capillaries and the filtration slits that are in, that are between pedicels, and the pedicel is a component of the podocyte. And the filtrate includes water, glucose, amino acids, ions, urea, many hormones, vitamins B and C, ketones, very small amounts of protein. So there's a list. And this list should be, part of something that you guys know. That, the fluids in the body and in the blood, what's in blood besides erythrocytes and, so red blood cells, white blood cells, all the different blood cell types that we've talked about. But there's also things that you need to grow. There's nutrients. And what are the nutrients? Well, sugar. And why we talk about glucose, it's a simple sugar, a common sugar that's used quite a bit. So glucose and amino acids are the building blocks of proteins. Ions are the electrolytes. Urea is a waste product that we're often trying to get rid of at a fast rate. Many hormones are going to be in there. Why are hormones in there? Why are they going to be part of the filtrate? Because they're small, right? They're small molecules, so they're gonna come through also. And a lot of these things you don't want to filter out. I mean, you don't want to get rid of your nutrients. You don't want to get rid of hormones per se. So you're going to want to reintroduce those back to the blood. And so that's what we were talking about, all that additional plumbing. Beyond the glomerulus that is filtering out the blood, these things are in the filtrate, but you want to put these things back except for the urea. So vitamins B and C, why B and C? Why not A, D, E, and K?
Water soluble.
Yeah, these are the water-soluble vitamins, so that's why. Ketones, very small amounts of protein, especially smaller proteins. So that's why they have in this diagram larger proteins. See, like if this was a folded 3D tertiary structure of a protein, even that is not going to fit so easily through this combination of fenestrated capillary and slits created by the pedicels. Okay, so after you filter, you want to reintroduce these things back into the blood. So that's why you've got these things. Now, why would you have thinner tubing versus thicker tubing? Well, thinner tubing, you're going to have less of a cell layer. So it's just going to be a single cell layer. If you slice into that, what does that look like? Well, you see a little hole. And, there's like one cell that looks like it's kind of spread out to make that opening. So that's what, a really thin tubule is going to look like. And the thicker tubules are going to be, you're going to have more cells around them. And therefore, different filtering properties. So you'll see that in the picture when we go back to that. Anyway, so here's the kidney. What about the blood supply part of this? So, here we've got an artery, and it's feeding, it is in this nephron in the glomerulus itself. But what does that part of the plumbing look like? And that looks like this. And they have a whole bunch of names for this stuff. But you're going to have the renal artery, right, which is the big artery going into the kidney. It goes in at this dimple here, which is the hilum. And these arteries, they typically segment out, and they're getting smaller as they branch off, of course. So they have a bunch of names for them. Interlobular artery, arcuate artery, as you're going around, interlobular artery, afferent arterial. So the arterioles are, of course, smaller, right? So you're branching down into smaller and smaller vessels. And so you see the way this looks. The renal artery is coming in larger, and it's branching out, and it's separating down. And this is where all of those nephrons are. And then we just looked at the liquid part of the plumbing, where we had the loop of Henle. But here we've got that overlaid with the blood vessels that are involved. And so this diagram makes a lot more sense, because we were just talking about the filtration of these impurities, quote impurities, but urea from the blood, and then we have these other nutrients, and we want them to reabsorb. So what do? Is we create a lot of plumbing here, and then we've got these small blood vessels, and these peritubular capillaries, and the vasa recta. These are associated with the loop of Henle. So these are all of these fine capillaries whose job it is to take back up those components of the blood that we didn't want to filter out. We didn't want to take out the nutrients. We didn't want to take out the vitamins and the hormones. We can reintroduce them. We can also reintroduce water, depending on whether the body needs to conserve water or not. And that's where all of this takes place. So then, once it's going around this, then this, of course, has got to come back into the circulatory system, so it's going to come back through veins. So then you've got the interlobular vein and the arcuate vein. Interlobular vein. So it's basically a parallel system, of course, of arteries to go around these nephrons, and then a vein system that have the same names. You see the names up here: interlobular artery, arcuate artery. So we've got interlobular vein, arcuate vein. So the parallel set of veins bringing the blood back out, and then this is the renal vein. So it's just plumbing, but it's pretty impressive, and I like the fact that the cell is able to negotiate molecules, and we exist on a level that we don't interact with molecules much. As people, we deal with a macroscopic world, but the cells live in this microscopic world, and actually a molecular world, where they're dealing with molecules, and they're moving around molecules and modifying them and getting the ones that they need. So here's a diagram of what that looks like. I'm not sure that this is a great diagram, but they do include this one other cell, the mesangial cell. And so we can talk about that. But there's also the podocyte and capillaries up there. So then the EM picture of this diagram. So if you were looking at this EM picture, what would you guess it would be? If I showed this on an exam. Well, firstly, it's an EM picture because it's black and black and white picture, right? And you see that the resolution is very high. And you're trying to figure out, well, what is all this stuff? Spaces are, lumens of some sort, but this is on a very small level too. So these are very fine openings. So turns out that this is a cell. So these are cells in this picture. And so this is the kidney, but E is an erythrocyte. Remember how they're disc-shaped? I think it's kind of a cool picture because it relates back to what we discussed about blood cell types. And you see them here. There's a lymphocyte here in this capillary, and it has both the podocyte. So the podocyte is up here, PD, and the like here, I guess P podocyte. But the pedicels are these fine-looking invagination things that were kind of a small microcilia looking. Don't think of them as that. I'm just trying to say that they're not that. But they are these slits, filter slits that are formed. So on EM they look like that. That's what these slits look like. It doesn't look like anything else in the body. So you guys should be able to identify that. And these terms, are critical for understanding how the kidney works and how urinary tract overall works. So the mesangial cell, that's down here. And why is that different than the podocyte? Well, it's a cell that kind of, it's one thing that they say about it is it supports things. Whenever you don't have a real defined function, you say, well, it's a supportive role. But actually, cells are doing a lot to provide different functions for each other. And one thing that this does is it's between these capillaries, so the mesangial cells are forming a support, and they're also sealing kind of the edges in, so that's a useful thing to do. They have a contractile function, so they see this little process here. You can actually see that there's some little thing coming off of this, and it's able to pull, so it's able to contract. It's like a mini muscle type thing. It adjusts contractions in response to blood pressure changes and help to maintain optimal filtration rate. That's kind of wild, isn't it? On that level, you're looking at just single cells here, and an electron micrograph of a couple of cells, and you see all the players here. It's kind of a elegant picture in that regard. You see some major players in the capillaries, and then the podocytes and the pedicels here, which have a structure unlike anything else, and those mesangial cells that have these processes, and are able to actually pull on the capillaries. And actually, these processes can help clean out the capillaries. Apparently, that's one idea. So that's kind of a useful function too. But depending on the blood pressure, they can open up the capillary or close it down a little bit more. So the taking away of the protein aggregates adhering to the glomerular filter, including antigen-antibody complexes. So, yeah, you're dealing on the molecular level, but these things can gum up. Remember, we were talking about circulating antibodies in the body, and if the antibody binds to an antigen, then it's forming a complex, and you can actually get a larger complex to form, an aggregate. And the aggregate could be coating the inside of the capillary and could actually gum up the capillary, and that would have a detrimental effect on the efficiency of the kidney overall, of its filtering system. So this is actually working on a microscopic or molecular level to pull out some of these aggregates. It's one of the common things that it will come across are the antibody-antigen complexes. And then, just by the way, this is typical of cells, It also secretes several cytokines and prostaglandins, other factors for immune defense and repair. So this is an important support cell. What do we know about cytokines, by the way? How is a cytokine different than a hormone?
Cytokines are the proteins. It's a proteins. Well, cytokines aid in inflammation.
In what?
Inflammation.
Okay.
They help with the immune cells.
They help what?
They help with the immune cells.
Immune? Okay. Okay, so cytokines are molecules also. But they have receptors typically on the cell surface, and a lot of these are receptor tyrosine kinases. So a cytokine is a signal on a cellular level that tells the cell to grow, for example, to divide, to be mitotic, or to make other factors. But a hormone is circulating in the blood. So, there are some cytokines in the blood, but hormones, steroid hormones, for example, is there a receptor for a steroid hormone?
They just go into the plasma membrane.
Yeah, they just go in through the membrane. See, that's quite a bit different. So, once they're in there, though, there's molecules that they can bind to. So there is a receptor, but it's internal. Since the steroid hormone is able to go through the plasma membrane, it can become intracellular, and then it can bind to its receptor there. Where does it go once that happens? Hormones binds receptor, intracellular receptor. Where does that signal go?
The nucleus.
What's that?
Nucleus?
Yeah, it goes right to the nucleus. It sits right on top of DNA, and it controls the transcription, the copying of DNA into RNA transcript. That's pretty direct, right? Cytokines is a receptor on the cell surface, and then there's a second messenger system, and you get into this larger network of effects, which are important as well. But I just want to give you some idea of, how these things are different. So the cytokines tend to be more localized effects. So we're saying that the mesangial cell is producing some cytokines, and it's a support cell, and it's sitting next to these other cells, the endothelial cells that make up the lining of the capillary. They're sitting next to these podocytes, and they're contributing some cytokines to, help define the way these cells should respond on a cellular level, as opposed to hormones that are circulating body-wide. You're trying to get, massive effects kind of immediately. You're not even dealing with a receptor on the outside of the cell. You're pervading right into the cells. So it's quite a bit different. So the resorption part, there are ions, so there's going to be ion pumps, ion channels on a cellular level. Transporters, enzymes, carrier proteins, these are all molecular type things. So, the interesting thing about histology is that we are somewhere between growth physiology and cell biology, and we're trying to pull this picture together. And then when you start talking about individual molecules, it seems like we've gone too far in the micro level. But it is important to understanding how the overall structure works and why that makes sense. So the kidneys are responsible then overall for this balance between secreting the filtrate and reabsorbing some of the things that you want. For example, like the amino acids, you don't want to lose your nutrients. You also don't want to lose things such as bile salts, creatine. What about drugs? So we medicate ourselves when we notice something's wrong, and so we ingest a drug. Is usually what we're doing. Can be injected sometimes, but drugs in the body is not used to dealing with drugs. So it's something of our creation that we've interceded and we've added something to the body. There's a phenomenon called drug clearance. So how long after you take a drug is the drug going to be pulled back out? And so the kidney, the urinary tract system is going to be a part of that. Because if you have a filter set up, a lot of the drugs are going to be diluted out and secreted in the urine. So there's drug clearance is an important aspect for modern medicine when you consider kidney function and the filtration of the kidney. You want to know about what the clearance rate is for the drug. So it ends up being pretty important in that way. Okay, let's look at this epithelia that we were talking about. So we said that dealing with the kidney, we're thinking it's a lot of epithelia. And then here's a picture, lots of epithelia. And I was commenting on what the epithelia looked like. This looks like a lot of other pictures, doesn't it, of some of the internal organs. But what's different? Well, for one thing, these tubules that are part of the nephron. Remember, we were saying that they have different thicknesses. They can either they can be thin or thicker, and then what does that look like? So here you have the proximal and distal convoluted tubules. Proximal, these guys, and distal. So the distal, they look kind of constructed of more of these epithelial cells. You can kind of see a circle of the epithelial cells around them. These guys also, but you don't see the individual cells right on the edge of the lumen like you do here on the distal tubules. So the proximal convoluted tubule was the one that was right after the renal corpuscle, which is got the glomerulus in it, and the distal is the thing coming out after it's gone through this lots of different plumbing with the nephron loop, the Henle loop, and it's coming back out. That's the distal one. But a slice of this kidney shows you these different structures. So that's kind of characteristic of kidney. So you guys would be able to identify this? Maybe not so easily, huh? Let's look at another picture. Cross-section of the renal medulla. Let's look at that. It's probably a little bit more fair, something like that, actually. Here it is down here. How about that one? Does that look better? So the structure that we were talking about before in the medulla was the medullary pyramid, saying that it looks like a fruit sliced open when you take a cross-section of the kidney, and the medulla has these compartments, and the pyramid is just a way of describing the fact that it's wider towards the cortex, and it narrows as it gets to dumping the urine into the ureter. But here, on this picture, we have loops of Henle here, that's labeled with a T, and it's very thin there. It has thick ascending limbs, so the ascending part of that tubing looks like this, the things with the A, like here's another one. And it also has collecting ducts, these guys. So that's a pretty good picture. What about that one? Satisfied with that? Would you be able to identify that as kidney? I mean, so keep in mind, kidney, you've got a lot of, 1 million nephrons. And a nephron is a lot of tubing. And the corpuscle is the ball that contains the glomerulus, and that's where the blood exchange filtering is going on. But then you've got all this other plumbing for reabsorption, and so you're going to see in cross section of a kidney, and this is a cross section of the medulla. And if it doesn't say kidney, if it says renal medulla, you know what that is. It's kidney, right? And the reason why you're seeing these circles is because that's the plumbing. And these thin portions of the plumbing only look like a single cell. See how there's no cells around that? It looks like one cell kind of just, it's the minimum thickness. And why would you want to have minimum thickness? Well, because it's all about filtration. We're talking about exchanging molecules here. So you don't, there are times when you want to have the thinnest layer possible. And we had talked about this before, where the organelles for cells sometimes are concentrated around a nucleus, just to provide plasma membrane free of organelles in order to get the maximum kind of exchange. So here, the thin tubules that are part of the group of Henle look like that, and the thicker ones look like this. And so you can see that these are thicker. See, this is sort of classic. Look at these nuclei here. The individual nuclei all around the circle. And that's a thicker ascending limb, they call it, or a loop, to the duct. And then the collecting ducts. Look at the size, the scale of the collecting duct. Here you see, these are all epithelial cells. You see the individual nuclei. So it's kind of reminds you a little bit of the smaller one here, but it's bigger, right? It's just bigger. And so there's more cells, and material is going to be coming into that. The collecting ducts are also a complex network, right? And they keep feeding into each other and getting larger and larger. What do you think are properties of the of any kind of tubule that deals with this filtrate? What does it have to do? Well, it's going to deal with a lot of urine, right? Which is kind of nasty stuff. So, the surface, the apical surface of the epithelial cells that are in a collecting duct, they're going to have to start to be tough. And as you start to look at the ureter and the bladder and the urethra, I mean, it's all about being resistant to urine, right? So, I think this is a good picture. So I like this one. So I think something like this is probably what I would show, and expect you guys to be able to identify kidney, and also be able to say renal. It's either a renal cortex or renal medulla. And the other pictures are pretty similar. Like here's the micrograph of the medullary pyramid cut transversely. Similar, almost exactly the same. This guy. Same players, different picture, but it's a little bit larger. So maybe this is even better. So you see that these thin parts of the tubule are very thin, and the thicker parts have epithelial cells all the way around, and a collecting duct is going to be even a slightly larger scale with a larger lumen. So that's another slice of kidney. Okay, so how does all of this work together? Pretty simple system. Kidney, ureter, urinary bladder, urethra. And then this was the complex system that we talked about, the nephron. And there's a million of those decorated around mostly in the cortex, but also in the medulla. And this doesn't show the blood part of it. Remember, we also showed you that, where there's lots of capillaries to reabsorb some of the filtrate. So that's physiologically, that's the thing that we're dealing with. So this next picture here, this is also a collecting duct, but they say, well, okay, so there's principal cells which are ADH regulated. What's ADH again? What?
Antidiuretic.
Right. Yeah, hormone. Yeah. So so that's going to control water resorption, right? It's kind of a crucial thing. So the body is monitoring how much water there is and how much you need. So where is ADH made?
Posterior pituitary.
Posterior what?
Posterior pituitary.
Pituitary, yeah. Why is the pituitary so critical?
It regulates the body's. No, it doesn't. It sends out the hormones.
Yeah, it's not the only endocrine gland, but it's the master endocrine gland, and it's being fed by it's connected to hypothalamus, so it's in contact with the brain pretty directly, and so you're able to get some crosstalk there, and so that's one reason to have that. What's on the other side of that, of the pituitary?
Hypothalamus.
No, that's true, but anterior, posterior. There's another small gland.
Oh, pineal gland.
Yeah, the pineal gland. That's kind of a cool gland too. You guys know what that one does?
Melatonin.
What's that? Yeah. Melatonin. So it's going to control our day-night, active versus sleeping pattern. Okay, so the pituitary is controlling ADH levels, and the principal cells in the collecting duct are responsive to that. And this is what this picture shows. It shows the collecting duct, and these are the principal cells, and they're ADH regulated. So that's an important aspect of the kidney too, to understand that overall the process is going to, the overall levels of water are crucial, both for blood pressure and for just having water generally. And it's kind of interesting that the shapes of what does an organ look like? Okay, so this is a picture of the bladder. And one of the things that you would expect from the bladder is that it can contract. right? If it's recently been emptied, if you empty your bladder, then the bladder can compact down. And then how is it going to compact? It's going to fold on a tissue level. There's going to be folds. And so when the bladder is full, it's going to look like this picture on the right. And when the bladder has been emptied, it's going to look like this picture here in the center. And this is a lower magnification picture of this same thing. So remember we were saying about histological pictures that it's always important to consider magnification as one thing. If you don't see individual cells so much, but you see areas that look like this, well, this is a low magnification picture. And this is where you can identify, epithelia would be on the outside. So facing a lumen is an opening, any kind of opening. So a lumen, the epithelia is always going to face the lumen. So if I would ask you, in a picture that you didn't recognize, where's the epithelia, you would be able to say, well, it's going to face the outside. Where's the outside? Well, the white part here, the lumen. The lumen, even though the lumen is the inside of the body, it's also where fluids are going through, so it's analogous to the outside. And the bladder has 3 layers of muscle associated with it. So this is what muscle cells look like at a low resolution, right? And one of the things about muscle in an organ is that you want to have it arranged in a couple of different ways because you want to squeeze, you want to be able to contract from several different directions. And so you have layers of muscle. And so on the bladder, you have, what kind of muscle is this, by the way? In the bladder. What kind of muscle is it? Cardiac, smooth, skeletal. Smooth, yeah. So, most of the time in internal organs we're dealing with smooth because we're not controlling consciously our internal organs, generally. They're going about their business, they're contracting when they need to, we don't need to think about it, it's all smooth muscle stuff. So, IL, ML, and OL are the inner, middle, and outer layers of smooth muscle. So inner being the closest to the epithelial, middle, and outer. And you see that they're all oriented slightly different ways. See the way this is kind of tangential at the middle layer, and this is a real cross-section on the outer layer, and then this is somewhat in between in the inner layer, a smooth muscle. So, so the way the histological picture looks is not always. Something can look drastically different and be the same organ. I kind of like these 2 pictures for that reason. Why is this picture blue? And this one's not.
Different stain.
It's a different stain, right? And it's one of the reasons why we set up the 2 staining stations in this class with the hemotoxylin eosin and the trichrome. Because the blue stain is for a lot of matrix protein. Proteins are, some of these proteins are staining blue, but it's epithelia that's staining blue. A lot of times on other tissues, connective tissue would stain blue because there's a lot of collagen. But there's protein that's staining blue on this epithelia. Is this, besides being an epithelia, it's a specific type of epithelia, a urothelium, right? For urinary. So urothelium, what did we say was unique about a urothelium again? As opposed to an epithelia, it has a unique thing it has to deal with, which is urine. So what does it need to do?
Tough top layer.
Yeah, it's going to have a tough top layer. So this trichrome staining here of the urothelium is kind of indicative of some unique proteins that are in here to make a tough epithelia that will be resistant to urea and so the whole urinary tract needs to have a urothelium. So so this picture is with a different stain, but it's the same same tissue, and it's just because this one is contracted, so you've emptied the bladder, and here the bladder is full. And you've stained with two different stains. And then this picture is just low magnification. A slice where you don't see individual cells. See, what do you see for individual cells? Usually nuclei, right? That's what you're used to looking at. When you see that spot, that's a nuclei, so that gives you an orientation. Oh, okay, so a cell is everything, it's the nuclei plus a bit of cytoplasm surrounding that cell. So what is the total magnification? Usually a couple hundred here. Whereas here, you're down below 100. Sort of think of it in that terms. Those terms. Yeah, they had another picture of something that looked a little different on the urothelium. That's here. Whereas this is the lumen, and then this would stain darker. I don't know, that's just an artist rendering, but if this is staining a little bit darker, the urothelium, and then there's the lumen. And these are cross-section of the ureter. So where's the ureter again? Catherine? Where's the ureter? In relation to the kidney? It's a tube that comes right after the kidney. Here we go. See? I showed you right there in the picture. So, how many kidneys do we have? 2. 1, 2. What happens if you get rid of a kidney? What's that? Yeah, you can survive with one. The kidney will, the one you have will enlarge and you'll get by. I know a guy that gave his brother a kidney. And he was always bringing it up after a few drinks, so he said he could drink more if he hadn't given his brother the kidney. Okay, what about, so this was the ureter. What's this down here? Yeah, that's the urethra. That's the bladder. So the urethra looks like this. Why does it look like that? What do we see here? Well, we see lots of epithelia. And what kind of a mesenchyme is that? Looks pretty densely packed. Packed with what? John, what's that packed with? Urethra. This is you're controlling your
Muscle.
your urine. Yeah, it's muscle. Right? You want to be able to control discharge of urine. So,
Sphincter.
What's that?
Sphincter.
Yeah, there's a sphincter that will close off too, but the overall flow rate. So this is all going to be muscle as opposed to connective tissue. So that's what an epithelia with a lot of muscle looks like. And then this is special epithelia. It's a urothelium, very resistant to urea. Okay, so that's enough, I think, for urinary tract. Take a look at it. It's chapter 19. And with some remaining time that we have, you guys have a choice to look at some slides, kidney, and we can also just kind of chat about what these sections look like versus the digestive tract, for example, respiratory tract. So we can just have a kind of an informal flashing of pictures up here while you guys can look at some slides. So, okay.